Hello friends, and in this very specific video we are going to talk about DNA chips, second alternative method of knowing DNA sequencing. First one was spiral sequencing. We already have talked about it. So let's see what is DNA chips. So first of all we are going to see its introduction, then techniques of it, and after that applications of DNA chips, and in the last we are going to talk about future of DNA chips. So let's see its introduction. So DNA chips or DNA microarrays are recently development for DNA sequencing as a result of advances made in automation and miniaturization. So a large number of DNA probes, each one with different sequences, are immobilized at defined position on the solid surface, and this surface is made up of either nylon or glass. Now you would have the confusion what is probes. Then probes can be complementary DNA, or you can say synthetic oligonucleotides for the preparation of high density arrays. Oligonucleotides are synthesized in situ form on the surface of glass or silicon. This results in oligonucleotide chip rather than a DNA chips. So it's a basic introduction. Let's see its technique. So I have diagrammed about it, and you just need to follow my lead. So it's a label DNA, and we are going to know about its sequence. What sequence does it have on it? So it's you can see it's a test molecule, or you can see label DNA. So it is your high density arrays, or you can say oligonucleotide chips, or you can say DNA chips. So a DNA chip carrying an array of different oligonucleotides can be used for DNA sequencing. So for this purpose, a labeled DNA test molecule whose sequence is to be determined, okay, applied to the chip. What I have said, that whose sequence is to be determined is applied to the chip. I have applied it. Now let's see, hybridization will happen on this chip, because its a test molecule will have complementary sequencing of on microchip, or you can say DNA chip. So it will hybridize, and hybridization occurs between the complementary sequence of the test DNA or oligonucleotides of the chips. So the positions of these hybridization oligonucleotides can be determined. These are the dotted point, that is the hybridization point, and these point can be determined by confocal microscopy. That it is confocal microscopy, and you can see dark position that shows hybridization signals. So each hybridization oligonucleotide represent an 8 nucleotide sequence that is present in the DNA probe. The sequence of the test DNA molecule can be deduced from the overlapping between the sequence of the hybridizing oligonucleotides. You can understand it. I know it's so much confusing, but you need to know that it is the hybridizing point, dotted point, and you can see it by confocal microscopy, and you would get the signal like a dark spot, and by this you can deduce the DNA sequence of our labeled DNA or test DNA molecules by hybridizing oligonucleotides, or you can say overlapping nucleotides. So these are our overlapping, you can say AGTC, CCC, and TTGGC. These are our hybridizing sequence. So it is the DNA sequence of your test molecules. So it is pretty basic. You don't need to be confused in it. We have taken a DNA and it would have complementary uh, oligonucleotides on this chip, and they would get be hybridized, and we can see these hybridized signal by confocal microscopy, and we can deduce our DNA sequence by overlapping oligonucleotides, and these are the overlapping sequence, then it would be the DNA sequence of our labeled DNA. So it is pretty basic. Now come to the applications of DNA chips. So we can detect the gene responsible for inflammatory disease that can cause the inflammation in our body. Also we can identify of gene that responsible for the development of nervous system. Microarrays for almost all gene for E. coli and yeast Saccharomyces cerevisiae. We have microarrays for almost all gene for these E. coli and yeast Saccharomyces cerevisiae, and we also can detect and screening of single nucleotide polymorphism, or you can say SNPs. And besides this, expression of several genes in prokaryote has been identified by the use of DNA chips. So these are your applications, and in future it is expected that within the next few years such chips will be available for complete genome arrays of eukaryotes too. Complete genome, then this will help the biotechnologists to capture the functional snapshot of the genome for eukaryote for higher organism. So it is the future, or you can say advantage can be higher organism or eukaryotes too. And the major limitations of DNA chips at present is that unavailability of complete genome arrays for higher eukaryotes. We don't have structured DNA chips for eukaryotes that could be available in upcoming years, so it will be advantage for higher organism. Now you can get the DNA chips or microarrays. If you have any comment, then comment down below. So thanks guys.